Okay, so now we're going to get into eukaryotic cells. And eukaryotic cells are the type of cells that we have. And what's going to make a eukaryote is that they're a little larger, and they also are going to have organelles. So what we're going to do is kind of follow through. If you look at this picture here, we're going to kind of go through all of these different organelles and talk about the different jobs that they have. And the way that we're going to do it is we're going to talk about the formation of a protein and how that kind of travels through the organelles of the cell. And then we'll talk about some of the extra ones that do different jobs. So that's kind of how we're going to go through it. So we're going to start out with this big purple thing right here called the nucleus. So you probably learned about that in science before. And so the nucleus is going to be where the DNA is stored, and it's also going to be the control center, of the cell. And so what that means is that it is going to actually power the cell and control everything that's going to happen inside the cell. So here's a closer view of it, and you can see here we're showing the surface of the nuclear envelope, which is the outer membrane. And so since we're showing the surface, that would be a scanning electron microscope picture, right? And then if we wanted to get a little bit more detail of what it looks like inside, then we use that transmission electron microscope picture. So hopefully you can see that difference there that we were talking about before. Okay, so going to make this a little bigger. Here's our nuclear envelope, and inside of it, inside the nucleus, is going to be what's called the nucleolus. And so that's going to be the real center part, and that's going to have its own jobs too. So now let's look at the jobs. So nucleus, like I said, is the control center, and it's going to be surrounded by the nuclear envelope. And the nuclear envelope is going to have pores that are going to allow some things to go through and other things not. Then inside, you're going to have what's called chromatin. Chromatin is going to be your DNA wrapped around proteins, and we'll learn about that in a later chapter. And so that's going to be how your DNA is kind of stored when your cell is kind of in like its normal mode, not when it's getting ready to split or do something like that. So the nucleolus is that like center part of the nucleus I showed you, and that's going to be where ribosomes are going to be created. So ribosomes are going to be really integral organelles when we talk about making a protein. Oh my God, and look what's next. Ribosomes. Isn't that amazing? Okay, so ribosomes are going to carry out what's called protein synthesis. And what that means is they're going to read the DNA, and they're actually going to create a protein as a result of that. And don't worry, we have a whole chapter to spend on that process. So your book's pictures of ribosomes are pretty funny. Here's a close-up picture of a ribosome. It has a large subunit and a small subunit. And so if we go back to our picture of the cell, all the little dots that you see everywhere, those are going to be ribosomes. So you're going to have two types of ribosomes. You're going to have free ones that are just kind of floating around in the cytoplasm, and then you're going to have bound ribosomes, and those are going to be the ones that are attached to this next organelle called the ER. Okay, so what's going to happen is the free ribosomes that are just kind of floating around, those ones are going to make proteins that are going to be important in that cytoplasm. Cytosol is the same thing as cytoplasm. Bound ribosomes are going to be attached to the ER, which is called the endoplasmic reticulum, and those are going to actually make proteins that are usually going to leave the cell and go somewhere else. Now, an important thing about ribosomes is that any cell that's going to make a lot of proteins, such as your pancreas, those are going to have more of those ribosomes because ribosomes are used for protein synthesis. So that's what you want to remember about all these cells, is that depending on what the cell's job is, it might have more or less of certain types of organelles. Okay. So the next one is going to be the endoplasmic reticulum, which totally sounds like you're really intelligent when you say a big long word like that. But we'll shorten it to ER. That's how a lot of textbooks will talk about it. And so if you look at the way the ER looks, it's all this blue stuff here. It's basically membrane that's kind of folded back and forth on itself. And so the ER is going to be really important in the process of protein processing. So you can see that there's two types of ER. You can have what's called smooth ER, which is going to be this little section that's kind of sticking up here, and then you have rough ER. The difference is that the smooth ER does not have any ribosomes bound to it, and the rough ER does. That's going to affect what job that they have. So here, if we look down here, the smooth ER is going to make lipids it's going to help with making carbohydrates, and it's also going to help with detoxifying things. So you could say that testes and ovaries, which make a lot of lipids, which are hormones, right, would have a lot of smooth ER because that's their job. Your liver is a big site of detoxification, so your liver is going to have a lot of smooth ER as well. Now, your rough ER is going to have the ribosomes on it, and so what it's going to do is it's actually going to make proteins. That makes sense, right? Ribosomes' job is to make proteins, and so the rough ER is going to help them do that. So then, if we look at our picture, we just talked about the nucleus here. Then we traveled out a little bit further to our ER. So inside the nucleus was where the ribosomes were being made. Now the ribosomes are out here on the ER, and they're helping us to make that protein, that chain of amino acids, right? Now that little polypeptide that we're following is going to end up here at the Golgi apparatus. And that's that kind of greenish membranous stuff that you see there. So what's going to happen is the Golgi apparatus is kind of like the Federal Express of the cell. What I mean by that is that the Golgi apparatus is actually going to take in that polypeptide that's very rough. It's going to package it up, and it's going to label it so that it can be sent to whatever part of the body that that protein needs to go to. So it's just like Federal Express. So it's going to have two ends. It's going to have what's called the cis face. That's going to be the receiving end. So that's where the polypeptide is coming in. And then it's going to have what's called the trans face, which is going to create little vesicles, which you could think of like the little FedEx trucks. And they're going to carry that in like a little bubble over to where it needs to go next. And usually that's going to be leaving the cell. Okay, so what's happening is inside the Golgi apparatus is modification. So that means that the protein is just getting processed and kind of fine-tuned a little bit more, and then it's going to get labeled with the phosphate or whatever type of signal it needs to go to the next place, right? So that's basically going to be how a protein is going to form. So starting in the nucleolus, where we're making our ribosomes that can read the DNA to make the protein, and so on. So if that's a little intense, don't worry. We're going to get into that a little bit later. Okay, now what we're going to finish up with are going to be some accessory organelles that just kind of do different jobs. So we've got lysosomes next. So if we look at lysosomes, I think I've actually got a better picture. Actually, there's our ER, and then the next one should be our Golgi apparatus. Here's our lysosomes. Now, lysosomes are really important because what they do is they're actually going to break down things in the cell. And so they're going to have a very low pH in there, very acidic, and that's so that they can break things down. Now, it could be food coming into the cell, and they're trying to break that down into its components. It could also be if you have an old organelle, the lysosome will actually fuse with it, and it will actually break that down so it can be recycled. So the point of a lysosome is that it's a digestive organelle. It's breaking stuff down. And that's a really important thing because what's going to happen is if you had all that acidity just anywhere in the cell, that could really harm the cell and break the cell components down. So the lysosome is going to actually be an area where the cell digestion can happen without harming the cell. Now, what's going to happen is the lysosome, here's our cell. The lysosome is usually going to situate itself near an area where there's a couple of H pluses hanging around. And so what that's going to do is inside this lysosome, it's going to use up hydrogen because it's breaking stuff down, and it needs to maintain that acidic environment inside the lysosome. And so what it's going to do is it's going to hang out in an area where there's a lot of H pluses, so it can pull those in to maintain that low pH. Okay? So one thing that was in that picture I just showed you on PowerPoint is a process called endocytosis. So you can see here you've got a piece of food and it wants to come into the cell. So what the cell's going to do is its like little membrane here is going to form like a little pocket, and eventually it's going to completely enclose that piece of food and make a food vacuole, which is like a little bubble that can travel somewhere else. So that's going into the cell, so that's a process we call endocytosis. Okay, and we will get into those other ones in a later chapter. So, vacuoles. Vacuoles are going to be something that you can think of as like a storage locker for a cell. Plant cells usually have really big vacuoles right in the middle. And so what the point is of a vacuole is it's going to store different things. It could store food for later, it can store water, it can store toxins, and you've actually experienced vacuoles if you've ever cut an onion open. And I will show you what you're thinking about. Okay, so we've got an onion cell right here. What's going to happen is that onion cell is going to have two vacuoles like that. And these two vacuoles are going to contain different chemicals. Separately, they don't do anything to each other, obviously, because they're separated. But if they are to break and touch each other, then they're going to form a compound that's going to make your eyes tear and hopefully make you not want to eat the rest of that onion. It's a defense mechanism. So what happens is when we cut through an onion cell, we actually cut those vacuoles and allow those chemicals to mix, and that's what makes our eyes tear when we cut open onions. See? Now I've even ruined onions for you. You're welcome. All right. So like I said, there's a couple of different types. There's food vacuoles. There's also contractile vacuoles, which are pretty cool. So those, let's see, you're going to see those in the cell lab. And so sometimes you've got a cell, like a little protist, a little organism, and it's going to have itself as a cell, and then it's going to have what's called a contractile vacuole. And what can happen is if a predator is coming near them, they can put water into that contractile vacuole and then contract it, and the water's going to shoot out, and then they're going to shoot in another direction so they can get away from a predator. So that's pretty cool. And then that central vacuole was going to be what was in this picture here, and you can see that with this plant cell. And a central vacuole it's like a storage locker. It can do all sorts of different things. Okay, the next one is a really important one. That is my dog itching. Sorry about that. And it's going to be these little jelly bean shaped things here called mitochondria. So here's a close-up view of mitochondria, and these should be your best friend because these are keeping you alive right now. Super important. So you can see that they've got an outer membrane and then they have an inner membrane. And so when we get into chapter 9, we're going to get into way more detail about why this is set up like this. And what happens is electrons are basically being passed from one protein to another in here, and that's what's keeping you alive. Basically, when you die, that little electron transport chain that's happening stops. That's the big difference. So that is going to allow a process called cellular respiration to happen. And so in order for a mitochondria to work, it needs to take in oxygen, and the byproduct is carbon dioxide. So cell respiration is why we breathe in oxygen and we breathe out CO2. Okay. The next type of organelle is going to be one that only plants have, and that's going to, well, as far as eukaryotic cells go, and that's going to be these guys called chloroplasts. And chloroplasts are going to be the green things that are going to allow them to do photosynthesis. So they are going to contain chlorophyll, which is going to allow the process of photosynthesis to happen. Chlorophyll is just a pigment. And the way that they are going to be set up is, you can see how this looks like little green poker chips. Those poker chips are called thylakoids, and those thylakoids are going to be in these stacks called grana. So one of them is a granum. In Latin usually means singular, and then when you end with an A, that means plural. So grana would be the plural. So that's going to be how those are set up. Another really important one is going to be peroxisomes. And peroxisomes are going to be important because those are going to detoxify alcohol. So I'm sure a lot of college students are thanking their peroxisomes after they've had a night out on the town, right? So here's a little peroxisome here. And so once again, if you think about like your liver that does a lot of alcohol detoxification, those liver cells should have a lot of peroxisomes in them. And when your peroxisomes don't function, that's when you can get poisoning and stuff like that happening. So the reason it's important to understand all these organelles is because in later classes you're going to actually learn all these different syndromes that can go wrong if one of those organelles isn't working. Okay, So those are all the parts of the cell. The last part is going to be talking about the actual cytoskeleton, which is going to be the inside of the cell. And we're just going to finish this up real quick. So it's kind of cool because if you think about the way that this picture looks, it's not really showing you that cytoskeleton very clearly. But if we look in a picture down here, this is more like what the cell really looks like. So it has a whole skeleton inside of it, and there's going to be 3 components that do different things. This is a cool picture where the cytoskeleton has actually been fluorescent, so you can see the cytoskeleton. But we'll go back to this little drawing here. So the cytoskeleton is awesome because it can actually be disassembled and then rebuilt so that the cell can move and do all sorts of crazy stuff. So there's going to be a couple of things that will help it to do that. Actin filaments are going to be ones that are going to allow the cell membrane to actually move so the cell can almost crawl. And so those in the picture here would be like along the bottom here. Then you're going to have microtubules, and microtubules are going to kind of help to support the cell. And they can actually be broken apart and put together in different ways. And there is actually a protein called dynein that can actually connect those microtubules and allow them to bend and flex. So if I've got these 2 things here, these are both microtubules, dynein would be the protein that would hold them together and allow them to do that, where they can kind of bend and flex. And then you've got your intermediate filaments, which are going to just be stable ones right in the middle of the cell. So those would be like these little yellow ones. So the blue rods here would be your microtubules. Okay, so those are going to be the different components of your cytoskeleton. Very, very important for keeping the cell's shape and allowing it to do all sorts of different things. Now, there are going to be what are called centrosomes, and centrosomes are, let's see, this right here, those 2 things that you see right there. And I've got a close-up right here. So a centrosome is going to be made up of 2 centrioles. This here is a centriole, and this here is a centriole. And a centriole is going to be composed of microtubules. It's actually composed of 9 sets of triplet microtubules. You see how there's like little groups of 3 here? And so these are important because they resist compression in the cell. So they allow the cell to maintain its shape, and they're also there for when we do mitosis and we need extra microtubules, they can kind of move around and leave a little trail of microtubules that are important. Okay, now one thing people don't realize is that there is also stuff going on outside the cell. So if you look here, you can see that there's all sorts of stuff coming off of the outside of the cell. And that's called the extracellular matrix. Very important because it's going to do a couple of important things for the cell. One thing is that since animal cells don't have a cell wall, they're kind of flimsy like that. So if they have that extracellular matrix on the outside, it's kind of holding them out and allowing them to have their shape like that. Another thing is that it allows them to kind of adhere with one another and form different types of junctions. And that's what we're going to finish up with here, is there are a couple of types of what are called intercellular junctions. So those are going to be junctions between different types of cells. So in plant cells, they have what are called plasmodesmata, and those are going to be channels that allow the cells to actually pass information from one to the other. So if like this hand is a cell and this hand is a cell, my 2 middle fingers connecting here, that would be the plasmodesmata, and that would allow information to pass. And I think I've got a picture. Oh, yes, I do. There we go. So this big thing is going to be a cell, and then this big thing is a cell, and then these little things in between are plasmodesmata. Then the next one are going to be tight junctions, and tight junctions are going to be like what you have in your skin cells here, and very, very tightly pressed together cells, and that allows you not to lose a lot of moisture to the environment. So like your epithelial cells in your skin are going to have tight junctions. And there is a picture of a tight junction right here. So here's a cell, and then here's a cell, and then in between is that tight junction. So very, 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 very tight together. The next one is going to be desmosomes. This is in animal cells. And these are going to allow your cells to fasten together and hold on really tightly in strong sheets. And so you're going to find these in muscle cells. Yes, sorry. So there's a desmosome right there. And then the last one is going to be what's called a gap junction. And a gap junction is going to be just like the plasmodesmata in plants, except it's in an animal cell. So gap junctions are going to allow information to pass through, and you find a lot of gap junctions in your heart cells, your muscle cells, and in developing embryos. So those are going to be all the parts of the cells, and we will talk about different parts at different stages of the semester, but it's really good to kind of see them all together and see how they work. Now there is another video that I'm going to post that's a tour of an animal cell and a tour of a plant cell. Very, very good videos that I definitely think you should watch, and it'll kind of help you put it all together.